Hello everyone, and welcome to the lecture that's happening while I'm away. So what I want to do today is cover some of the major important events in the history of life that have sort of had a profound effect on the way that the evolution of life has unfolded. And we can think about this in the context of other things we've been talking about. So we spent a good deal of the first part of the semester thinking about microevolutionary processes within populations, thinking about how selection, drift, gene flow, mutation cause evolutionary change within populations. Then we sort of pulled out from there to think about how things like differential adaptation because of divergent natural selection in different populations can lead to the evolution of reproductive isolation and lead to the origin of species. And we've just recently started thinking some about phylogenetics, which helps us construct the history of relationships across life and helps put some of these things we've been thinking about in more of a historical context. What I want to do now is really dig in more on that historical context and think about how specific events can play a role in evolution on Earth. And I want to start with one of the big ones that I'm actually not going to talk about too much, but that is the origin of life, which we think happened about 3.5 to 4 billion years ago. Now, I'll just point out first that I think your book does a really nice job of covering the origin of life, and I'm not going to go into all the details the book does, but I just want to kind of couch in some context some of what the book's talking about and give us sort of something general to think about. So the basic thing I want to think about first is sort of how did life arise from non-life? And the short answer is we don't really know. There are a number of different models and hypotheses that have been put forth, and some of them seem to be mostly consistent with what we know at present, but none of them is definitive. So I'm going to talk about one of the more popular models and sort of use that as a stepping stone to thinking about the origin of life. And what I want to talk about in specific is the Oparin-Haldane model, and this model basically posits that the Earth itself possessed all of the necessary ingredients for the origin of life. In other words, the Earth had everything that was needed for life to arise. And this is basically, this model posits a series of steps that would be necessary to go from non-life to life. So let's think about those steps to get from non-life to life. All right. So the first key step is that we have to have a way to get the non-biological, in other words, without living things, we need a non-biological process by which we can synthesize organic molecules, simple organic molecules, such as amino acids and nucleotides. If I can spell molecules and such as amino acids and nucleotides. So we need a way that we can go from non-living to basic building blocks of life in terms of these simple organic molecules. And then the model basically posits that from these organic building blocks, these organic building blocks were assembled into biological polymers. And by that I mean things such as proteins or nucleic acids. That is basically strings of these basic building blocks. And then the sort of third step posited by this model is that some combination of biological polymers were assembled into self-replicating organisms. And that these organisms, and these essentially fed off of the other organic polymers out there. Now, so how does all this work? So one of the first things to think about is what were these supposed first replicating things? And the answer is there are good reasons to think that the first replicating things were RNA. So these first self-replicating organisms were likely RNA based. The reason for this is twofold, or two related things. That is that RNA has properties that in some way mimic DNA, and that in some ways mimic proteins that make that RNA a very good candidate for being the sort of original life form, or the basis of the original life forms. And those are that RNA can store information, which is the same thing DNA can do, right? So DNA stores information, but it's important to point out that RNA can store information, but not as well as DNA. And the other sort of key component of the basic molecular machinery of life is to catalyze reactions. And it also turns out that RNA can catalyze reactions, but not as well as proteins. So in other words, RNA can, right at this point, we have our DNA for information storage and proteins for catalyzing reactions. But RNA can do each of those things, not as well as DNA or proteins, but it can do both of those things. So we have an idea of what the first replicating organisms might have been. Can we get from non-life to RNA using only sort of ingredients and conditions that exist on Earth? And the answer is that it's hard to say. So we know that there are a number of experiments have been conducted, which you can read about and should read about and hopefully have read about in your book. These experiments show that there are plausible whereby each step in the Apr-Haldane model is feasible. In other words, experiments have showed that each of these steps, each of the individual steps is feasible, but not necessarily that the combination of steps together is feasible, although one could argue that the individual steps being feasible implies that the entire process would be feasible. And these experiments also sort of support the idea of RNA being a potential original life form. But that doesn't mean that those things actually happen. This is one of the difficulties of the historical aspect of trying to understand things like the origin of life, is even though one can show that a series of events is possible or plausible, it does not mean that those things actually happen. So there is support for the Apr-Haldane model, whereby you have non-biological processes leading to the synthesis of organic molecules, those organic building blocks being assembled into biological polymers, and then those biological polymers being assembled into self-replicating organisms, particularly in the form of RNA, but that doesn't mean all those things happen. So it's all plausible, but this does not mean that the Apr-Haldane model is correct. Doesn't mean it's incorrect either. It simply means we don't know at this point. And there are other possibilities and ideas out there. So this is probably what I would consider the most standard model. But as you've certainly read about and thought about, even in the context of the reading quizzes, there are other hypotheses that suggest that life may have been brought in from other planets, such that life may have actually arisen elsewhere and then come into Earth during the early ages of the Earth, around again 3.5 to 4 billion years ago. So there's a lot really to be done still, and this is really a sort of an open question, which is one of the reasons why I don't want to dig in too strongly in any specific way on it, but rather sort of give you an idea of one way in which these things could have happened, and again with your book providing you a little bit more details about it. But the main thing I want you to take home from this is that we have an understanding of how you can go from non-life to life. We understand specific steps that could be involved, and we have experimental evidence that each of those steps is at least plausible. And we have an understanding of what the first organisms might have been, but none of that necessarily means that is actually what happened. Now, at this point, I want to show you a short 5-minute clip with one of the leading experts on this idea of the RNA world, Jack Stovstack, talking more about this RNA world hypothesis. In other words, this idea that RNA was the original basis for life.
The general organizational structure of modern cells is very complicated in the sense that it's highly self-referential. So every aspect of this process, the central dogma, the transmission of information from DNA to RNA to proteins, and then down to building structures with function. Every part of that depends on all the other parts. So for example, the replication of DNA requires DNA, but it also requires RNA and proteins, the polymerases. The transcription of RNA requires DNA, which is where the information is stored, but it also requires many proteins, and it also requires many other RNA molecules. And similarly, the formation of proteins occurs on a remarkably complex machine, the ribosome, which itself is composed of RNA and proteins. So for decades, it was very hard for people to think of any reasonable way in which such an internally self-referential system could emerge spontaneously from a chemical environment. And the answer to that really came from thinking about RNA and the different things that it can do. So this simplification in thinking came from the realization that RNA can not just carry information, but can also catalyze chemical reactions. And that realization led immediately to the hypothesis that in primitive cells, RNA might be able to catalyze its own replication, also carry out biochemical functions for the primitive cell. And so then all you really need to think about is a cell with RNA molecules encapsulated within some kind of primitive cell membrane that itself could be a self-replicating structure. So the history of this idea actually goes back to the 1960s. And three very smart people, Leslie Orgel, Carl Woese, Francis Crick, hypothesized, in part on the basis of the complex folded structure of tRNA, that an early stage of life might have evolved RNA as the sole macromolecular basis of evolved machinery. And so this lets you think of simple cells emerging with just a single biopolymer, RNA, and then later on, as evolution developed more complex cellular structures, information storage became specialized in DNA, and most functional activities became specialized as the job of proteins. Now, although these ideas were put forth in rather elementary form in the 60s, and of course nobody took them seriously at the time, because there was absolutely no experimental evidence for the idea that RNA could catalyze chemical reactions. At the time, people had just started to get very detailed, high-resolution information about how proteins catalyzed reactions. And the idea that a molecule like RNA could do the same thing seemed ludicrous. So it wasn't until almost 20 years later, with the work of Tom Cech and Sid Altman, and the experimental demonstration that RNA molecules could actually very effectively catalyze at least certain types of chemical reactions, that people took this whole idea of an RNA-based early stage of life seriously. And so that hypothesis, the RNA world hypothesis, was really, the ideas were summarized by Wally Gilbert in an article in 1986. And this has really become the foundation of a lot of thinking about early stages in the emergence of life. Okay, so apart from the basic fact that RNA does and can catalyze chemical reactions, is there any other evidence that early life might have been based or exclusively on nucleic acids. And in fact, there are several lines of circumstantial evidence. One of them is the structure of many cofactors. So here you see acetyl-CoA, just one example. But the working part of the molecule is the thioester out here. And for no obvious reason, there's a nucleotide at the other end. And really, the only way to make sense of that is that the nucleotide is a handle, either a relic of primitive ribozyme or something that was easy for primitive ribozymes to grab hold of, and thereby using this cofactor catalyze reactions in a thioester-mediated way. Now, there are other examples. Here is vitamin B12, another very important catalyst. Its working part is this complex corin ring. But down here, you see, again, a nucleotide. What's it doing there? It's probably another relic of the RNA world when all of this complicated biochemistry was being catalyzed by RNA enzymes. Yet another example is the very way that the substrates for DNA synthesis are made. And they're not made de novo, as you might expect if DNA came first. They're actually made from pre-existing ribonucleotides. And so the transformation of ribonucleotides to deoxynucleotides is catalyzed by the enzyme ribonucleotide reductase. And this unusual synthetic pathway can be viewed as the relic of the fact that early in time, metabolism and RNA synthesis used ribonucleotides, and only later, when DNA was invented or evolved, was there a requirement to make deoxynucleotides, and so they're made from the closest available substrate. Okay, finally, perhaps the most important and dramatic piece of evidence for the early role of RNA in primitive forms of life is the actual structure of the ribosome. And so this is a slide from Tom Steitz showing a view into the active site of the large subunit. So this is the peptidyl transferase center. And this little green structure in here is a transition state analog that marks out the place in this gigantic machine where the chemistry is happening. And what you can see is that it's these gray squiggles, which are the RNA, that completely make up that active site. So all proteins are generated by an RNA machine, the RNA central region of the ribosome itself. So again, this only makes sense in terms of an early stage of biochemistry dominated by RNA functions, which then over time evolved the ability to make proteins which are now so important in all modern biochemistry. So if we want to...
So the origin of life was clearly a fundamentally important and key aspect in terms of the historical events that have led to the diversity of life we see now. And in fact, we don't even know at this point whether the origin of life occurred once or many times. It is indeed likely that most of the life, if not all life that we see today, has a common origin, but there's possibilities that life had various false starts through time before taking off as it has. But what are some other sort of key innovations and radiations that have had sort of major effects on the overall patterns of biodiversity we see? In other words, these macroevolutionary patterns. And along those lines, one thing I want to point out is it is fundamentally true that rates of diversification, by rates of diversification, I mean, in essence, rates of speciation minus rates of extinction. That is, the rates at which you accumulate lineages over time. So rates of diversification are not constant over time. Rather, you see evidence of key innovations, that is, the evolution of new features that really drive adaptive radiations. That is, periods when you have an increased rate at which diversification is happening as as species are evolving to occupy novel niches that perhaps didn't even exist prior to that key innovation. So what are some of these key innovations? Well, one of the clear ones is the evolution of multicellularity. So multicellularity, that is, life forms that are having more than one cell, likely evolved about 3 to cellularity, likely evolved about 3 to 3.5 billion years ago. In fact, the evidence suggests, just from the basis of interpreting things on a phylogenetic tree, that there must be independent evolutions of multicellularity at least 46 times. So multicellularity is not one of these things that has evolved once, but has evolved many, many times. Multicellularity has many potential advantages. The large size of a multicellular organism gives them a competitive advantage over single cellular organisms. And it permits complexity by allowing for differentiation or division of labor, where some cells in an organism can specialize on subset of functions such as some aspect of growth or reproduction, where other cells specialize for other important functions. And I'm not going to talk more about multicellularity now, but as you are aware of it by this point, I assume, are reading a paper for this week's think piece that gets at the evolution of multicellularity in an experimental context. So we'll have a chance to talk more about that during the think piece discussion on Friday. Another sort of key in a period of innovation or of radiation that I want us to think about is what happened around the onset of the Cambrian. So prior to the Cambrian period, there was very, very little evidence of diversity in terms of life relative to what we see now. But by the Cambrian, we already had a rich diversity of multicellular life by the time we get to the Cambrian. And in fact, by that period, we had ancestors of all extant phyla. And perhaps, although this is not as strongly believed as once was, perhaps in ancestors to representatives of phyla that no longer exist at all. And I'll show you an image real quick here. Where did it go? So here is one of the more famous representatives from the fossil record of the Cambrian period. So this is the Burgess shale fauna, and the dates to the Cambrian period, so about 515 million years before present. And here's a sort of artist rendering based on fossils of the diversity of organisms you see. Some of the key things that are going on by this point is you have a greater number of ways of making a living than were available before. We see sort of most the ways of making a living, most of the niches existing that exist in the modern world. And again, the sort of diversity of phyla we see in the modern world, and as well as some rather wacky looking organisms. This one in particular is pretty cool. So this creature right here, its actual limbs are these little things down here. But for a good 20 or 30 years after this little guy was discovered, people thought that these were its legs and always drew it as this thing walking on stilts. But in fact, that's not the case. So we have the Cambrian explosion around 515 million years ago. And we see the appearance of all of our modern phyla. And I'll actually have a question for you about the Cambrian explosion in just a second, but I want to talk about a couple more things first. So let me come back here. So that's sort of one of our key events. We see this radiation of all modern phyla, making all of the livings that we see today. Another sort of major thing that happened was the colonization of land. Colonization of land. And this happened with arthropods about 450 million years before present. And it happened with plants about 420 million years before present. So both of these major groups of organisms colonized land about 450 to 420 million years ago. And this set off a couple things. One, land was a brand new niche for the most part, totally unoccupied. So there's all these different ways of of existing on land that weren't there in the oceans. It also began a long history of plant insect interactions and coevolution. Where you see insects evolving to feed on plants in numerous ways, often specializing heavily on specific species or genera of plants, and plants often taking advantage of insects as well, and for things like pollination. Pollination, not pollinization. So this was followed later by colonization of land by tetrapods. So tetrapods didn't colonize land until about 360 to 395 million years before present. And the sort of current famous fossil that is thought of as a pretty good idea of roughly what the organisms that were right at that transition into moving on land might have looked like is referred to as Tiktaalik. Tiktaalik. And I'll show you a nice little picture of Tiktaalik here. So here's Tiktaalik. So again, it's sort of morphologically and physiologically, it's a nice intermediate between tetrapods and fish. It had scales and webbed fins. But like a tetrapod, and really more specifically like a crocodile, it had a flat head with eyes on the top of the head. It had an actual neck. The fins of Tiktaalik were part fish fin and part tetrapod limb. On the outside, they looked like fins with webbing. On the inside, though, they were clearly more tetrapod-like. So again, colonization of land by tetrapods, another sort of example that led to major radiations. I want to point out, though, that you can also get adaptive radiations on smaller scales. So we've been talking about really major innovations that led to evolutionary radiation. But you can have evolution of novelty on smaller scales that can lead to smaller radiations. Examples include things like if a species of insect, say a butterfly might colonize and adapt to a new family of host plants, and that might allow it to open up with many new opportunities within that family of host plants that might lead to a rapid burst of speciation. You also see adaptive radiations often on islands, for example, in island birds, such as the sort of famous Darwin's finches, where you have a species colonizing in essence, what is a somewhat open environment with many niches to be filled that can allow for radiation to fill those niches. So with that, I want to pose a question for us to think about for a second. All right, so here's the question I want to pose, and I realize that it's unlikely that you're sitting with a group to actually discuss this question. So instead, I want you to sort of spend a few minutes and think through it yourself. Preferably even write down an answer yourself so that you really have sort of thought through the question. So the earliest evidence of animals in the fossil record comes from the Ediacaran faunas, which is earlier than the Cambrian period, earlier than the Burgess Shale. So here we're talking 565 to 544 million years ago. The Ediacaran animals were small, asymmetrical, or radially symmetrical, whereas we have, by the Burgess Shale fauna, by the Cambrian, we have bilaterally symmetrical species. Again, ancestors of all modern phyla, including arthropods, vertebrate echinoderms. As I mentioned, the Cambrian explosion not only constitutes a remarkable increase in the diversity of morphological forms, but of ecology too. So you had sessile filter feeders. In the Ediacaran, you had basically sessile filter feeders or predators that floated around consuming plankton. By the Burgess Shale, we have highly mobile benthic and pelagic predators, filter feeders, grazers, scavengers, detritivores. One explanation for this increase in the number of ways to make a living during the radiation is it was triggered by an environmental change. In particular, we know that oxygen levels increased dramatically about 543 million years ago, so basically right at the end of the Ediacaran, and made higher metabolic rates and larger bodies possible. Do you think increased oxygen levels are a sufficient explanation for the increased ecological diversity of the Burgess Shale fauna? Are there other developmental or ecological explanations that are possible? So take a few minutes, think through this on your own. All right. On Monday, I will briefly poll the class on this question as well as another one I'm going to ask in a little bit, and we'll talk about them as a group because I think that'll be more productive than me just answering right now. So we'll chat about this question as well as another one that's coming up in a second on Monday. So we've talked about how you can have key innovations that can drive adaptive radiations and sort of an increase in rates of diversification. And next week, before I get into human evolution, I'll talk a little bit regarding what we know about that in the context of evolution of development and sort of genome evolution. But what I want to talk about at this point is sort of the flip side of that, and that is mass extinctions, which are another set of events that have major, profound effects on the overall patterns of biodiversity, sort of macroevolutionary patterns. So the first point I want to make is that extinction is the ultimate fate of all species and clades, that is higher groups of organisms. So everything will eventually go extinct. All species go extinct. The vast majority of species that have ever existed are now extinct. It's the ultimate fate of all things. With that said, extinction rates vary through time, and there have been basically 5 periods of very high extinction rates. And these are what we often think of as mass extinctions. So let's look at an image of that real quick. Here's an image actually from the text looking at the 5 sort of big mass extinctions. So on the y-axis here you have the percent of families, not species, but families that went extinct during that period. The different times are shown here on the x-axis, time in millions of years, and you'll note these sort of 5 big blips highlighted in red here, where you have very, very high rates of extinction relative to this background rate. And so the presence here, deeper in the past, is here. So I just want to talk about a couple of these briefly. So the first one I want to talk about is the Permian extinction. So this occurred about 252 million years before the present. It is the largest mass extinction ever. So during this mass extinction, we lost 90% of marine life and 70% of terrestrial life. So this was a big deal. We don't have a perfect grasp on what drove this. Suggested mechanisms include one or more large bolide impact events, so impacts from objects from space, massive volcanism, coal gas fires, explosions from the Siberian traps, a runaway greenhouse effect triggered by sudden release of methane from the seafloor due to methane dissociation, or methane-producing microbes. And there's also potentially more gradually contributing things such as sea level change, increasing anoxia, increasing aridity, and shifts in the ocean circulations driven by climate change. So a number of things are potential contributors. Almost certainly some combination of these things are what drove this massive extinction event that shot up the extinction rate, resulting in this massive loss. Then there's the famous one that everyone sort of knows about. That's the one at the Cretaceous, what is now called Paleogene, and what you may know as Tertiary. at the Cretaceous, Paleogene, or Cretaceous Tertiary boundary. So this what has been called the KT extinction, but that doesn't make as much sense with the current use of words. This occurred about 66 million years before present. This is when we lost 75% of all species, and this is when we lost non-avian dinosaurs, which is why this one's so famous. This is one we maybe know much more about. So it's now generally believed that the Cretaceous Tertiary or Cretaceous Paleogene extinction was triggered by a massive comet or asteroid impact, and the catastrophic effects that unfolded because of that on the environment, including a lingering impact winter that would have made it impossible for plants and plankton to carry out photosynthesis. And we actually have a pretty good candidate for where that impact happened. It's in a crater called the Chicxulub crater off the Gulf of Mexico that was identified in the early 1990s. That's a good candidate for being the actual site of that. So now that brings me to the next question I want us to think about, that again I want you to think about some now, and then we'll discuss when we get back together on Monday. And that is that ammonites and nautiloids were affected differently by the Cretaceous Paleogene mass extinction. Both groups were mollusks with buoyant chambered shells that lived in open water habitats. Ammonites went extinct during the Cretaceous Paleogene extinction, but nautiloids survived. The two groups, interestingly, had different reproductive strategies. Ammonites likely produced many free-swimming young each year that fed near the ocean's surface and grew rapidly, whereas nautiloids produced just a few large eggs each year, each of which rests in the depth for up to a year before hatching into small, slow-growing organisms. Based on these different strategies, I want you to come up with a hypothesis to explain why nautiloids, but not ammonites, were able to survive an asteroid impact. So a hypothesis that would explain why this different would happen, and an experiment that you could have conducted to test this hypothesis if you had been around before the asteroid impact drove one of these groups to extinction. So take a few minutes right now to think through this question, come up with an answer, sketch something down even so that you'll have something to talk about and remember this question when you come in on Monday, and then we'll talk about it then. So I'm just about ready to wrap up here. I want to end with a couple key points that I really want, that I mean as the take-homes from everything we've just been talking about. And that is that mass extinctions and the evolution of key innovations, important new traits such as multicellularity, are specific historical events. Right? These are historical events. These things happen once or some sort of finite number of times. That's not a very good word. Events that can have substantial effects on macro evolutionary patterns. So we can have these individual events that can have major effects on the overall patterns of at the macroevolutionary level, looking at the diversity of life across the tree of life. So it is true that biological diversity is a function of the microevolutionary processes we have discussed. In other words, biological diversity is certainly driven by a product of mutation, selection, genetic drift, gene flow, or the lack of gene flow between between populations, and the evolution of novel species to the evolution of reproductive isolation. But it is also profoundly affected by individual historical events, which are generally going to be less predictable. So that even if we have a full understanding of these sort of microevolutionary processes, it may not be sufficient to explain all of the macroevolutionary patterns, because that requires having an understanding also of these individually important historical events, such as mass extinctions and the evolution of key innovations that set off totally new radiations. And with that, I'm going to wrap up the lecture. Friday, we're going to have the think piece like we were planning on the paper looking at experimentally thinking about the evolution of multicellularity. And then for the next 2 weeks, our last 2 real weeks, we'll sort of talk some about the evolution of development and genome evolution to sort of finish up kind of this module really that we've been working on, and then transition into human evolution, which is our final topic for the semester. See you all on Monday.